Hi everyone, good morning or good afternoon or good evening. Welcome to the last of the Pharma Research Network Research Methods sessions for 2023. These are led by the CRFS Research Methods Support Project, RMS. And I think we're on number 4. Yeah. So these RMS sessions are attempting to build on the Pharma Research Network working group meetings. So this one we're referring back to the last working group meeting from the 8th of November, where Elizabeth from the Manor House Agricultural Centre presented some very interesting background on the Manor House and some of the activities they're conducting at the moment. I think about 3/4 of the way through the video, she talked about the use of rapid experiments. And I'm going to talk a little bit more about the examples she presented. But we thought this would be a good opportunity to drill down a little bit into research methods relating to rapid experiments or studies. So I'm Jane. I think most people know me. I work for Stats for SD, so I'm with the Research Methods Support Project, and I'm based in Nairobi. Okay, so today in the seminar, we're just going to talk a little bit about what are rapid experiments. I decided halfway through putting together the presentation that I would call them studies, because they're not always the traditional what we think of as experiments. Why can they be a valuable component of our research toolbox for CRFS research, and in particular for pharma research networks? And then how can we do them well? If we're going to use them, how can we do them well? And when might they be the most suitable or appropriate to use? So rapid experiments or studies, they're well adapted to answering the types of research questions about what works or what doesn't, rather than our more traditional research hypotheses and trying to confirm scientific facts. The clue is in the name. They should be quicker than traditional research. Rapid experiment sits within this sort of broader concept of rapid research. And I think most of us being part of the pharma research networks will be quite knowledgeable on the rapid rural appraisal, RRA, which came about in the late 1970s because in response to some of the challenges with these large-scale structured questionnaire surveys, which took ages to analyse and didn't always answer the question. And it provided a sort of alternative technique for people to learn from local people about their realities and challenges. And then this led on to participatory rural appraisal, where I personally see a lot of links with our Pharma Research Network work, which focuses more strongly on facilitation, empowerment, behavior change, local knowledge, and sustainable action. So a lot of the principles that we use with pharma research networks. Rapid experiments and studies, they may be good enough to take the next decision, but they don't necessarily meet the sort of conventional standards of scientific rigor that we would think about. But they can still have a role to play. So what are they? They're standard practice in many industries, actually, both in research, but also in the private sector, in product development, in large-scale manufacturing, when in the R&D departments of these companies. So I found one definition of it, which was rapid experimentation is an agile approach to the product development process. With this approach, frequent experiments are deployed in an attempt to discover new and innovative ideas. Experiments can range in severity from simple A/B tests to larger field experiments. But it's not without its critics. Again, across the private sector, there's been a lot of critique about some of the, particularly Silicon Valley or the new tech companies. Well, I say new, but they probably came up 30 or 40 years ago. And I found one that was a review that said the methodology of rapid experimentation focuses on discovering problem solutions through leap of faith assumptions that will delight the end users. So I think they were trying to say that it's a very attractive method, but that perhaps it has its limitations. But I think we can avoid some of these challenges if we're applying it in our FRN research by using it when it's appropriate and with good designs. So why might it be a useful approach for our CRFS FRN research? Well, it does link well to our FRN principles, particularly around farmer participation, and you'll see later the research that is rigorous and useful. It can complement and add to FRN farmers' own testing. When I was preparing this presentation, Rick mentioned that in the soils group discussions, they'd been talking about learning experiments done by farmers, which help them to learn something, but that it may contribute little to the more general scientific knowledge. But I think with minor adaptations, collecting a bit more data collection, being a bit more systematic, we could learn something that also advances the science as well as being useful for the farmers. It can also provide us with additional research of agroecological technologies to support adoption at scale. Again, in another meeting, Dave from FIPS was presenting about how they trained 27,000 farmers in the soil method to control fall armyworm, and they'd conducted over the last 3 or 4 years several different surveys of subsets of farmers to see what was happening, to see whether they were using it, whether they'd experienced fall armyworm, and how effective they were finding the soil method. And at the time when Dave was presenting, he was saying that they wanted to look at whether farmer characteristics, potentially sort of socioeconomic status or soil types, might influence the uptake and effectiveness of the soil method. So they did observe some quite interesting results, but the sample of their farmers tended to be quite unbalanced, with few observations like certain soil types or certain different types of farmer. It was difficult to come out with any clear conclusions. And again, with some minor tweaks, maybe aiming to balance the sample of farmers, they could get much stronger data to answer some of their hypotheses. So as I say, I think it can be a useful tool in our methods toolbox. But we do have a dilemma. Yeah. So over the years, we've always thought about, if I'm going to evaluate a practice, I need to test it over multiple seasons. We've got the options by context. I need to try it in different environments. And we know that we don't get a full picture if we just look at the first observations or just in one context or one season. Rapid experiments obviously is not designed for that sort of multi-season or multi-context, whereas our long-term more conventional research is. But also we are working with farmers who need quick answers, and that was also mentioned by Manor House, and these first observations can be informative. So this is where rapid experiments could be useful, but obviously the long-term experiments require more data. So how do we balance the advantage of being sure, but the need to get answers quickly and not waste resources? For example, in our case, farmers' time and land. Yeah. So the answer, or the proposed answer, is that we use well-designed rapid experiments to studies when it's appropriate and when it's reasonable enough to get good enough answers. It's not replacing more conventional research, but it's adding to our toolbox of methods. So if we're going to conduct rapid experiments or studies, how can we ensure they're good? Yeah. So the usual principles of good research design, which we've talked about in other sessions, still apply, and that we use when we're designing our experiments or our research. Objectivity. Do we have clear hypotheses or research questions or objectives of what we're trying to achieve? Replication. Are we not just going to observe something one time, one place, and draw some conclusions? Randomization, which could mean different things depending on the type of study. So it might mean, taking random samples of farmers or other types of randomization. Validity and reliability, so avoiding the bias that we can generate. So biases can come from the researcher, They can come from measurement, the selection of farmers or the selection of treatments. So it's still avoiding that. Precision. So that's through the type of local control. So the types of aspects that we can do to improve precision are balancing, blocking, grouping, a bit like the FIPS example I just told you about in terms of the sample. And then also repeatability. If we try again, will we see similar results? And then another aspect of conducting good rapid experiments or studies is balancing the trade-off between our confidence in results and the time and resources it takes. So as I mentioned in our dilemma slide, we need to conduct the research quickly, but we want to evaluate over multiple seasons. We need to use resources efficiently, but we want to evaluate over different contexts. And we need precise measures of the results versus we need to use the results for the next step, not to establish a scientific fact. And another aspect of good rapid experiments is evaluating the study, just as we would any other research study. So collecting good data and measuring things that will be useful for both farmers and researchers. So I just want to talk a little bit about the Manor House examples. They provided one example in the video, and then also Elizabeth provided me with another one. So the first one is bokashi, so testing alternative ingredients. So they found, for those of you that haven't seen the video, I'm just recapping. So bokashi is working well for their biopesticide FRN farmers, but they can't afford some of the ingredients. Some of them are quite expensive and must be sourced off farm. So they were asking Manor House for substitutes that can either be from the farm or are cheap. And Manor House recognized that without adapting some of the bokashi ingredients, farmers were not going to adopt the technology and they would lose momentum. So they started with quite a classic, like an on-station design with two replicates for each of 3 to 4 treatments, where they kept one of them the standard recipe. They had one very simple indicator to measure, which was the pile temperature. Each experiment was very rapid. It took 14 days. And what they want to do next is conduct a nutrient analysis to see why different ingredients might be working, but also take it a step further to apply it to a fast-growing indicator crop such as cowpeas. So I'm assuming that the classic design in bullet 2 was randomized. I'm guessing it probably was. This is a good design. How could it be improved? Well, potentially involving farmers more in the evaluation. Perhaps there's a plan to do that, or perhaps they didn't do it and didn't notice and didn't mention it in the video. And the other idea is, so the next step, this applying to a fast-growing indicator crop, will that be rapid or not? It could be either. I could see an argument for why they could try both approaches. And then the second example, which wasn't mentioned in the working group meeting, I think, which was about consumer preference testing. So they have their farmers who could produce dried indigenous vegetables for sale in the dry season, but they weren't very sure whether there was actually the consumer demand for these dried vegetables. So Manor House conducted a study at both, it was an opportunistic type study where they were invited to a value addition of food processing exposition, and they conducted a study while there. And also, I think they followed up with some of their farm group meetings. So they delivered preference testing questionnaires and a number of dishes to taste. So consumers didn't know what they were testing. This is sort of controlled conditions. Elizabeth mentioned they tried to allow for the order of tasting, so not everybody received the same dish first or last. But maybe I'm not sure whether she conducted a formal design for that, but there are various formal designs we can use to ensure that that's balanced across all the respondents. I also don't know how they developed the questionnaires, but again, they whether they did it together with the farmers, whether they did it together with a pilot group, and what were the key measures they were doing. So they found some interesting results. The ones that Elizabeth presented was just from the exposition. And the outcome was that after 6 months, 4 of their farmer groups had been convinced that there's a market for the dry products and had built solar dryers and were growing crops to preserve. So again, it's an example of a good rapid design. Could it have been improved? So maybe, as I mentioned earlier, the formal design to ensure the balancing, maybe piloting the key responses. And I would have liked to see, although I'm sure it's there, a synthesis of the results across the tasting events and actually how you analyse and combine that data. Okay, so in the last slide, I talked about balancing the trade-offs. Well, actually not the last slide, 3 again. So rapid experiments are most appropriate when our research questions do not involve these trade-offs, and then it's an easy call. Do I do rapid or do I do more conventional? So, for example, in these controlled situations like the bokashi that Manor House had, or for screening options. Screening options, we haven't had one of those in the examples, but say in plant breeding, where you've got many varieties. In the first season, you might test like 60 varieties, and then you might actually screen out the ones that are just not working for then conducting follow-up. And also, I think it's particularly useful when the study complements other activities. So it's just one component of the action research. And again, I think the bokashi example is quite a good one because they've also had quite classic research on the bokashi as well. It's likely that there are, it's likely that there are opportunities to conduct rapid experiments and studies in most of our CRFS FRN projects. But the key is in identifying when they're the most appropriate method for us to use. And when might they not make sense? So Manor House has, they made the rapid experiments work because of their attitude, their drive, and their facilities. They have good resources. They have links with farmers, student interns, and connections to experts to consult. If assembling everything that's needed will take months, then aiming for rapid methods is probably not going to work. Okay, so just in summary, the rapid experiments and studies can be a valuable component of our FRN research methods toolbox, complementing more conventional research. They should be used where it's appropriate and applying the same good research design and implementation principles that we use for conventional research. They're likely to be most appropriate when complementing other more conventional research activities. And you're going to go into groups after this, and I think, well, you are open to discuss anything. I think it would be useful. It would be really nice if you also talked about examples from your own projects of rapid experimentation, how these are working or how they worked. And then also about whether for your ongoing research, there are aspects of research rapid experiments or studies that could be an option. And we will share the feedback on this, and we're ready to follow up with any support. Okay, so thank you very much, everybody. I wish you all the best for the holiday season, and we will see you early in the new year, hopefully.